Everyone. It's my great pleasure today to introduce a physician scientist who is at the forefront of advancing clinical treatments for genetic retinal diseases, Dr. Mark Pennesi, MD, PhD, to our Department of Ophthalmology Distinguished Speaker Series for his talk titled Novel Treatments for Inherited Retinal Degenerations. Dr. Pennesi is the new Director of Ophthalmic Genetics of the Retina Foundation in Dallas, Texas, and he maintains an appointment at the KCI Institute at OHSU. He received his BSc in bioengineering from the University of Pennsylvania, where he graduated summa cum laude. He then went on to the MD PhD at the Baylor College of Medicine before his residency in ophthalmology at the University of California, San Francisco. To complete his fellowship training, he went to OHSU, where he was a fellow in ophthalmic genetics, medical retina, and electrophysiology. He subsequently joined the faculty at OHSU, where he was promoted to associate and then full professor of ophthalmology at the KCI Institute, and was the professor of molecular and medical genetics at OHSU and the chief of the ophthalmic genetics division. Dr. Pennesi is a clinician scientist who is at the forefront of many first-in-human and first-in-class therapies for inherited retinal diseases as a PI or co-PI, and his clinical trials of note include those for RP65, ABCA4, ASH2B, CNGA1, and CNGB3 achromatopsia, X-linked retinoschisis, CEP290, and many others. He has won many awards, including career advancement and research awards from FFB, Alcon, and RPB, and serves as editor and on the editorial boards of journals such as BMC Ophthalmology, TVST, Ophthalmic Genetics, and ILVS. He's published many book chapters, mentored many students, and has published more than 150 peer-reviewed publications. Please join me in giving a warm welcome to Dr. Pennesi.
Thank you for that warm introduction. It's really a pleasure to be here today. Yesterday, I gave a little talk on kind of my career for the physician scientist, and I was going back through my papers, and I found my first paper that I published in graduate school, and my co-author was Chris Palczewski. And so it's important to get good collaborators, because then you'll have a very successful career. Also, Vladimir, I was trying to think back to when we met, and I think it was way back when we were both graduate students at a FASTSEB meeting. And so it's really amazing, the people that you get to know along the way progress and become leaders in the field. So I'm really happy to present some of the data today. And I think the one takeaway that I want you to have from this talk is that we are very close to being able to treat inherited retinal degenerations. And as a clinician, when I got my first job at the Retina Foundation at age 19, working with David Birch, there was nothing for these diseases. And for almost 20 years, there was nothing for these diseases. But that is now starting to change. Okay, these are my financial disclosures. And much of this work was done at the KCI Institute. So I'd like to acknowledge my team there, including Paul Yang and Leslie Everett, who are my colleagues. So I'm going to kind of go through several different diseases and several different trials. And I'll just kind of start off with a little bit of a background of inherited retinal degeneration. So there are descriptions of inherited retinal degenerations going back to the time of the Greeks, but the first person who's really credited with coining the term retinitis pigmentosa was a Dutch ophthalmologist, Frans Cornelis Donders. And, interestingly, he was a friend with Helmholtz, who invented the first ophthalmoscope. And so he looked in and saw patients with these pigment changes, and they had night vision, and he sort of identified retinitis pigmentosa. And we know now that that was a little bit of a misnomer. Retinitis implies inflammation. They thought this was an inflammatory disease. We know that really this is a genetic disease, and that it's a progressive retinal degeneration.
There's a second screen on here.
Okay. Okay, perfect. So we know now that there are really over 300 different genes that can cause inherited retinal degenerations. retinitis pigmentosa, upwards of 100 different genes. There are many different modes of inheritance. It could be autosomal recessive, autosomal dominant, X-linked, even mitochondrial. And this is rare, approximately 1 in 4,000, but not that rare. And patients often present with problems with their night vision, problems with dark adaptation. As time goes on, they start to have peripheral field constriction, and ultimately that affects the central vision. Most patients will go legally blind by the time they're in their 50s, and some will go completely blind. So this is kind of how the world of inherited retinal degeneration looks now, and it's kind of complicated, right? So we have 300 different genes, and there's a lot of overlap. You can see in the center, retinitis pigmentosa is the most common, but then we have these other entities. There are syndromic forms of retinal degeneration, such as Usher syndrome, where you have hearing loss, Bardet-Biedl, where they have kidney problems, extra fingers and toes, as well as cone rod dystrophies. And any given gene, can cause one of these entities, but there's also overlap where some genes in a certain patient might cause retinitis pigmentosa. In other patients, they might cause a cone rod dystrophy. So what can we do for these patients? Well, there's certainly some interventions that all patients can benefit from, such as low vision aids and treatment. I'm going to really focus today on gene therapy, but there are other methods of protection, such as neuroprotection, cell-based therapies, which is really exciting, artificial retinal implants. Now, the kind of intervention that we're going to give is really going to depend somewhat on the patient. So if we think about a patient with Leber congenital amaurosis, which is an early onset, very severe retinal degeneration, these patients lose vision very quickly. By the time they're in their teens, they may be legally blind. So it's a very rapid degeneration. Retinitis pigmentosa, on the other hand, often starts later in mid-20s, and it's a slower degeneration. And cone rod dystrophies fall somewhere in between here. So depending on where the patient is in the course of their disease, we may have different strategies. So if you're on the upper left hand of this curve, our goal really is going to be to preserve the cells that you have, to try to prevent the degeneration. And we might use gene augmentation therapy, gene editing, antisense oligonucleotides, or neuroprotection. But if you're down on the bottom right half of this curve, the cells are gone. So those preservative therapies really aren't going to work. At this point, we really need a restorative therapy, such as stem cells or optogenetics, artificial retinal implants. So what kind of things can we do for all patients? So, one of the most important things I always teach the residents is just a really good prescription for their glasses. A lot of patients come in and they don't have a good glasses prescription. You can actually really improve their quality of life by making sure they have the right prescription. But also, there are other vision aids, magnifiers, orientation and mobility training for many patients, guide dogs for some patients. And, the whole kind of tablet devices have really revolutionized, low vision, because most people now can use their iPad for magnification. So there's something called cystoid macular edema, which is very common in retinitis pigmentosa. So these are OCT scans. If you're not familiar with this is actually a kind of an optical way of imaging the retina. So we're looking at the retina in cross section here. And you can see on top, this patient has these big cysts in the retina. And we don't really understand why patients get cystoid macular edema, but it's actually quite common. Maybe a third of patients with RP can have cystoid macular edema. And the really important thing is that this is treatable. So we have drugs such as carbonic anhydrase inhibitors that we can give. And so you can see on the bottom, this patient was treated with a topical medication, and the cysts go away and their visual acuity goes from 20/60 to 20/25. So this is another intervention where we can actually improve their quality of life. Micronutrients. So, back in the 1980s, there was a lot of studies on vitamin A, and vitamin A was recommended for retinitis pigmentosa. More recently, there have been some publications sort of questioning whether or not vitamin A is really effective. And in fact, the original lab that did that research, they're not actually giving out vitamin A anymore. So we don't typically recommend vitamin A for our patients, but this is something you will still see in the literature quite a bit. We do recommend antioxidants, primarily things like DHA, so salmon, fatty fish, lutein. And then there's a lot of other research being done on things like N-acetylcysteine, which are antioxidants. So I'm going to move now into talking about gene therapy. I think most of you remember sort of the basic biological dogma that DNA codes for RNA, which codes for proteins, and that makes up the cells of our body. And if we have a mutation in the DNA, this can have different consequences. So, some mutations might result in no protein being made. Other mutations might result in a mutant protein being made that has toxic effects. And so the approach to fixing these is going to really depend on the situation. So if you have a non-functional protein, you're going to try to replace it. And we might do that with gene augmentation. Alternatively, there are things such as antisense oligonucleotides where you might be able to do splicing modulation. But if you have a mutant protein that's causing problems, then we need to turn that off. And gene editing is a good approach for that, as well as antisense oligonucleotides. So just to kind of go through, gene augmentation has really been the workhorse for gene therapy. And the concept is that you're missing the normal copy of the gene, you're not making the protein. So we take a normal copy of the gene, we put it into some kind of delivery vector. Most commonly these days we're using AAV, although people now are looking at other things such as lipid nanoparticles. And these go into the cell and put a normal copy of the gene into the nucleus. So there's another form of gene therapy where we can target the RNA. So once again, if we have a mutated gene and it's coding for a mutant RNA, we can create a complementary strand that matches up to that. That'll create a double-stranded RNA, which will get broken down in the cell and decrease the protein. So that's RNA silencing. Similarly, you can pair that with a normal copy of the gene. So not only do you decrease the bad one, but you replace the normal protein. So gene editing is kind of the new thing that is really exciting. And here, it's a little bit of a different idea. We have our mutated gene, and we're going to use bacterial proteins, such as CRISPR-Cas system, to make a cut. And then you can introduce a template to get homology-directed repair, which will then have a repaired gene. Alternatively, in certain situations, you can just cut out the portion that is mutated, and sometimes that will fix the problem. Kind of the next generation of that is base editing. And the idea here is you might have a single nucleotide mutation, and we can take a Cas protein that has been deactivated and then pair it with adenosine deaminase or cytosine deaminase and actually elicit a switch in the mutated sequence and fix the gene. And then sort of the most advanced method is prime editing, where now not only do we have a Cas protein, but we introduce a reverse transcriptase and a template, and we can actually edit an entire sequence. So as I mentioned before, AAV has really been the workhorse. And if we look at the wild type AAV, there are different parts. There's a capsid, and then there's a DNA component. The DNA component has two major genes, one that is involved in replication, the other the capsid, and they're sort of flanked by these ITR sequences. We strip out the rep and cap genes, put in our promoter and our gene of interest, and we make replication deficient vectors. We can then introduce these into the eye, and there are different ways to get them into the eye. So you can just inject them right into the vitreous, intravitreal injection. We can put them subretinal, or even more recently, suprachoroidal delivery has been attempted. And this is just a little video showing how the subretinal injection is done. So a standard vitrectomy is performed, and then a 41 gauge needle, which is a very thin needle, is used to create a bleb underneath the retina, and you can see the fluid here as it starts to expand. And this actually resolves usually pretty quickly within a day of the surgery. So these are all the different trials on the left that we were involved in at KCI Institute, and on the right are other trials that are occurring. And so you can see that there are many gene therapy trials going on in the eye. And we've used many different vectors, different AAVs, both subretinal, intravitreal. We've even done some trials with lentivectors. And so we now have a pretty good experience with understanding how these vectors work and some of the potential problems with them. So of course, the real seminal work with gene therapy started with RP65, and a lot of the credit goes to Gene Bennett and Appelstein, Al McGuire. And this is a disease that when you have a mutation in RP65, you get Leber congenital amaurosis. So these patients are not able to make the visual pigment, and they have profound night blindness. In addition, over time, they slowly have a retinal degeneration, and most of these patients go completely blind by the time they're 30. You can package a normal copy of RP65 into an AAV2 and deliver this subretinally. And this works. And the way they demonstrated that was by creating this novel test, which was a multi-luminous mobility test, where patients had to navigate a maze at different levels of light. And this is just a video showing how this works. And what you can see is this is actually a very dim level of light. It's a highly sensitive camera. So you see how slow the patient is, and they just stepped in a hole and they're barely moving. If we look at that same patient at the same level of light a year after gene therapy, I think you'll notice a little bit of a difference. So they're able to navigate quite easily. More recently now, we have long-term data from this trial that shows durability. So this is showing the improvement in multiluminous mobility over 5 years. And anecdotally, there are patients now who've been treated 10 years ago and are still having a benefit. So it looks like at least, this is durable for 5 to 10 years, which is very exciting. So this was approved by the FDA in 2017. It's quite expensive, $425,000 per eye. We've treated a number of patients, and I just wanted to show you a couple of our patients that have been very successful. So one of the first, this was a 4-year-old male who was diagnosed at 6 months with Leber congenital amaurosis. And when we saw him, his visual acuity was around 2100. And after treatment with Retinal, the visual acuity actually improved to 2030, which is quite amazing. And you can even see changes on the electrophysiology. So this is an ERG, which measures the electrical activity of the retina. And at baseline, you can see that this patient has flat ERG. There's no signal at all. And 6 months after treatment, you start to see the emergence of a signal in the cone ERGs. And by 1 year, you can actually measure a signal in both the rod and cone ERGs. So this is objective data showing that we were actually treating large parts of the retina. Now, there has been some issues with this treatment. And a few years after the start of treatment, there were reports of these focal areas of atrophy in some patients that were really occurring faster than what you would expect from the degeneration itself. And indeed, other groups have seen this. And so this parafoveal atrophy is something that we don't completely understand. We think it may have something to do with the dose being a little high. Fortunately, we only see it in about 10 or 15% of patients. And it actually does seem to correlate with the patients who have a greater improvement in sensitivity, seem to be at a higher risk of atrophy. And personally, I think this might be a little bit of overdosing. This is one example of our patients. So this was a 13-year-old male with RP65 retinopathy who was treated in both eyes with Retinal. Once again, you can see their initial visual acuity was 2400. 3 years after the treatment, they'd improved to 2100. And here are the images showing where the blebs were delivered. And this is a special test showing their visual fields. And so this is something called two-color dark adaptive perimetry. And what we do is we have the patient look into a big dome. There are little flashes of light. They hit a button whenever they see it. And if they see it, every time you get a signal. If they don't, it's black. And so you can see for this patient, for both the red and the blue stimuli, most of their sensitivity is dark. They don't really see anything out in the periphery. So following gene therapy, just 3 months after gene therapy, you see this huge improvement in their visual fields, especially to the blue stimulus. And by 1 year, this is really consolidated, and it actually correlates to where the subretinal blebs were delivered. And so this really does confirm that this works, and it works very well. Now, if we look at this patient, though, we do see that over time, they do start to develop these little areas of atrophy, and they do correlate to where the bleb was delivered. And once again, if we look at those areas, the sensitivity there is almost supernormal. And so my hypothesis is that we might be overdosing a little bit. Okay, I'm gonna shift over to another study. And this is a disease you may not have heard of, but it's called X-linked retinoschisis. And it's another genetic eye disease. And it's results from mutations in a protein called RS1 or retinoschisin. We don't really fully understand what retinoschisin does, but a lot of people kind of think of it as a glue for the retina. It's very important for synaptic transmission. And when you don't have it, the retina kind of splits apart, as you can see here. So there were actually multiple efforts to develop gene therapy for this. And one of the first was led by Paul Sieving at the NIH, and he used an AAV8 to do intravitreal delivery of RS1. And he saw, anecdotal success in one patient. You can see here that in the treated eye, the schisis disappears, temporarily. Unfortunately, it does come back with time. And what they found was as they went to higher and higher doses, they started to get inflammation, and the patients actually required significant immunosuppression. We were also part of a competing trial, and this was an industry-sponsored trial using a very similar approach. Rather than an AAV8, this used a modified AAV2 vector, also injected intravitreally. And I'll show you, here's the construct of it. This was the design of the trial. So it was a phase 1/2 dose escalation trial. And we had really good natural history data from these patients. So we sort of understood, how much of a change in different parameters we needed to really dictate what success would be. Unfortunately, when we actually went and treated 27 patients at 3 different doses, we really didn't find that many patients met any of these criteria. So it really didn't work. There were some anecdotal successes. This was one patient that I had in the high dose group. And you can see that in the treated eye, there was this resolution of the schisis, which was actually quite impressive compared to the untreated eye. But this was not common in the other patients. The other thing that we really learned from this trial was the importance of inflammation that can occur with gene therapy. And this was one of the first intravitreal deliveries of AAV. And we found that 4 to 6 weeks after the treatment, a lot of the patients were coming in with this picture where they had these are called KP or keratic precipitates on the surface of the cornea. These are immune cells. And you can treat them, they go away when you give steroids, but this is definitely an inflammatory response. The other thing that we saw were cells in the vitreous. So you can see the gel of the eye here, these little white cells. And these are a little more difficult to treat. And many of the patients had to go on to oral steroids to resolve this. So, at the end of the trial, we saw that, a large proportion of the patients actually did have inflammation. And as time went on, we changed the way that we were dealing with this. So this just kind of shows the low dose group. And at the low dose, we didn't really see a whole lot of inflammation, but at 4 to 6 weeks, there was some inflammation. Mostly you could treat this with topical. But as we went up in dose, most of the patients had inflammation, and we started to have to give people prophylactic oral steroids. This shows in the green here, this is the levels of anti-AAV capsid antibodies. And it seems that almost every patient will get an increase in these antibodies to AAV. Although, interestingly, they don't really correlate perfectly with which patients get clinical inflammation. And that was kind of interesting. We think that really the inflammation is more complex than just an antibody response. There may also be T cell responses. And in fact, 3 of the patients in the trial ended up requiring steroid-sparing immunosuppression. So we had to put them on methotrexate. So, I think this was a very important lesson in the sense that, you can have adverse events, and we still are learning. We're really at the very beginning. So what about future directions for X-linked retinoschisis? Are we done? And the answer is no. So the idea would be that, well, if intravitreal gene therapy is too inflammatory, maybe can we go subretinal? And there's a lot of apprehension for doing that because these patients have very fragile retinas. They can get retinal detachments. But there's a new kind of vector that may offer some hope. So when you look at traditional AAV vectors, when they're delivered subretinally, the transduction really is limited to where the subretinal bleb is made. But Shannon Boy at University of Gainesville has developed a variant where the vector actually will spread beyond the subretinal bleb. And so there is now a trial. We've started a phase 1/2 trial that is actually delivering a gene therapy vector for XLRS subretinally using this spreading vector. We've treated a few patients. I can't really say much more about that, but we're hoping that this might actually be an alternative way. Okay, the next disease I'm going to talk about, I think, is very exciting because this is probably or hopefully going to be the next approved gene therapy for X-linked retinitis pigmentosa. And X-linked RP is one of the more severe forms of retinitis pigmentosa. So most commonly, it's caused by mutations in a gene called RPGR, which is on the X chromosome. So the typical story would be a 7 or 8-year-old boy who comes in with problems with night blindness, decreased peripheral vision. And because it starts much earlier in life, these patients tend to have a much worse outcome. And you can see here, they can have quite advanced progression. Their visual fields can be quite constricted. Many patients will be legally blind by the time they're in their 30s. Interestingly, females can also be affected, but it's quite variable in females because it's on the X chromosome, and because of X chromosome inactivation, you can get skewing. So females can be completely asymptomatic with just some mild pigmentary changes to having very severe disease, almost as bad as what we see in males. So RPGR it's a pretty interesting gene because it has 19 exons, and the most common isoform that's expressed throughout the body uses 1 through 19. But there's a splice variant that is specific to the retina that uses exons 1 through 14, and then a big chunk of the intron, which is this OR15 sequence. And this is a highly repetitive sequence with GC, and so it's prone to mutations. But you have to design the gene therapy to include this particular splice variant, and you also have to do codon optimization so that such that when you express the gene therapy, you get the right product. And that tends to be pretty difficult, but a number of companies have done this. So, this is pretty amazing. We have 4 different companies right now working on gene therapy for X-linked RP. And so the first I'll talk about was an effort by Nightstar and Biogen. And this was using an AAV8 vector to deliver a codon optimized version of RPGR subretinally. And so this shows patients treated in the phase 1/2 trial. The red circles show where the blebs were delivered. And what you can see, there were 5 different cohorts at different doses. And in the first 2 cohorts, they really didn't see much of a change in sensitivity. So the outcome measure here is something called microperimetry, which is a visual field that looks at the sensitivity of the macula. When they got up to cohort 3, though, they actually did start to see some improvement. So if you look at these patients, this is sort of baseline. Normally, this image would be all green, meaning that the sensitivity was very good. Here, they're down to a very low sensitivity. And when you look at the treated eyes over time, going downwards, you can see, or I'm sorry, going across, you can see that there's sort of an expansion of the sensitivity and also an increase of the sensitivity. When they went up in dose, though, they really didn't see a change, and they thought maybe this was because of inflammation. So this just shows one of the patients who had inflammation. So you can see early on, there's an improvement in the sensitivity, then it decreases if you treat them, and you can see these subretinal deposits here. And then if you treat them with steroids, the sensitivity actually comes back. So, this teaches us that inflammation can occur with subretinal delivery as well. You might have an advantage with subretinal delivery, but it is not immune from it. Now, unfortunately, this trial went on to a pivotal phase 3 trial, and it failed to meet its endpoint, and the program was stopped. But there have been 2 other subretinal programs that are still moving forward. So one is originally from AGTC, now Beacon Therapeutics, using a very similar design. And there have been a number of different trials. So the first was a phase 1/2 dose escalation study. And this is looking at visual acuity. So if you look at the treated eye versus the untreated eye, this shows all the patients. These are spaghetti plots. You see that most of the eyes that were treated, there's actually an improvement in visual acuity as compared to the untreated eye. There's no improvement. Now, this is not enough to get approval. For the FDA, you really have to get a 15-letter improvement. And here we're seeing 5 to 10 letters. So it's hopeful, but it's not actually enough to show the significance that we need. If you look at the microperimetry on the top, you see once again a treated eye, and starts off very small, and then as time goes on, you see an expansion in the sensitivity as compared to the untreated eye, where there's really no change in the sensitivity. So we think the gene therapy vector is doing something. And if you look at all of the patients who meet this criteria to be a responder versus the untreated eye, there is a statistically significant difference. And what's really interesting is you can actually see structural changes on the OCT. And so this was an analysis done by my colleague, Paul Yang. And if you look at the OCT, there's a particular structure called the ellipsoid zone here, which really correlates with whether or not photoreceptors are intact. And after gene therapy, you can actually see a thickening of the ellipsoid zone as well as an expansion. And that really correlates with what we're seeing on microperimetry. So this is sort of a second independent indication that this gene therapy is actually helping the photoreceptors, and perhaps even the structure is improving. This has gone on now to a phase 2 trial where they randomized a lower dose versus a higher dose, and looking at microperimetry and other endpoints. And this just shows that they were well matched between the 2 groups. There was really no adverse events, a little bit of inflammation. But what I want to show you is, the improvement. So once again, if we look at a patient with a treated eye, we see a big improvement, expansion, improvement in sensitivity as compared to the untreated eye. Here's a second one. And statistically, once again, we see the high dose that 5 out of 8 patients had an improvement, whereas in the low dose, you really didn't see an improvement. So this is now going on. We just started another trial, which is we're treating the contralateral eye of patients. And then there's a pivotal phase 3 trial planned later for this year. Interestingly, there's another competitor, And same idea, subretinal gene therapy. This is from Mira GTX. They have a dose escalation study with a expansion cohort looking at randomized patients. They're using visual fields as well as microperimetry, and they too have shown improvement in their patients. So this is a completely different company, different vector, also showing improvement. And here are their microperimetry results, and the data is quite similar. And so, for me as a clinician, this is really exciting because when you see two different companies using different techniques, both getting the same result, I think that's very suggestive that something is working here. They are going on they've actually completed a phase 3 trial. They're actually using a little bit of a different endpoint, which is a mobility maze where patients have to go through this maze at different light levels. And they've shown that in their phase 1/2 trial that patients who are treated can actually navigate this faster. So hopefully by the end of the year, we should have the results from this trial. So I'm very excited. I think we may have something very soon. I'm not going to go into the 4th trial, which is an intravitreal delivery for RPGR. That's still quite early on, but that is another avenue. Okay, so achromatopsia, I'm going to talk about this trial a little bit. So this is another sort of rare inherited retinal dystrophy that you might not have heard of. But it's really fascinating because it results from mutations in proteins that are important for cone phototransduction. And in essence, these patients do not have functional cones. Their rods are still working, but because they don't have cones, they have low vision. They're typically visual acuity around 2200 or legal blindness. They're very sensitive to light because essentially they're using their rods for all of their vision. And they have no color vision because they don't have any cone function. This just shows the mutation. So the most common mutations are in 2 different genes, CNGA3 and CNGB3, which make up the cyclic nucleotide gated channel in the cones. And there have been several different trials, both for CNGA3 and CNGB3. I'm going to talk about one that we're involved with, once again, with AGTC. And this is using a subretinal AAV2 vector. 2 different trials, one for CNGB3, one for CNGA3, but I'm going to talk about them a little bit together. So, once again, sort of a classic dose escalation trial, but this is where things get kind of interesting. So when we treated these patients, we didn't really see a change in visual acuity. And that wasn't real surprising because, if you think about it, if you don't have high resolution vision when you're young, your brain may not wire up to have good vision, the visual cortex. Even if you fix the genetic problem, you may not be able to see it. But what we were able to do was to look at the sensitivity with visual fields. And so once again, this is one of these 2-color dark adapted visual fields. And specifically, this is using a red stimulus. And why is that important? Well, rods are not as sensitive to red light, so we're trying to select for cone function. If we look at the baseline in this patient, they really have very limited sensitivity. You kind of see a little ring here, and actually that's probably being mediated by the rods, because even though rods are not sensitive to red light, they do have some response. But a year after gene therapy, we see this big improvement in sensitivity, and it's right where the bleb is delivered. So this is one patient. Here's another patient. Once again, we see this boost in sensitivity. Here's a 3rd patient, and then a 4th patient. So, this gene therapy is actually doing something. Interestingly, the patients reported less photophobia. And that's actually the biggest complaint that these patients have. So unfortunately, we treated adults, got to the highest dose, things were going well. We then went down to children, thinking that we probably need to treat younger. And if we're going to get a visual acuity improvement. In fact, you may have to treat, at 1 year old to really get cortical development. When we went into the children at the same dose that we had been in the adults, we started to see severe inflammation, subretinal inflammation, and the trial was halted. So, once again, this is kind of an important lesson that you can see safety in adults, but when you go to children, the inflammatory response might be very different. And, we have to be very careful. And, this is why we spend a lot of time doing these trials. We start at low doses, but there are going to sometimes be adverse events. And even a therapy like this, where it does seem like, it has a functional improvement, adverse events can essentially end the trial. Just kind of looking a little bit at, why would you get inflammation and can you do anything about it? Certainly we know there are many different responses to AAV. And I think that a lot of people think about the capsid, and there are definitely pathways such as TLR2 that respond to the capsid, but also the DNA component within the capsid, I think, plays a very important role, and other receptors might mediate that, such as TLR9. And there are a lot of different things we can do. So really, the most important factor is dose. So the higher you go in dose, the more inflammation you get. When you get up to 1E12, you are almost certainly going to have some inflammation. So, for the basic scientists out there who are really designing these vectors, it's really important to have something where you can have the dose down below 1E11. Rarely do we see inflammation at 1E10 or 1E9. So if you can sort of improve that, functional effect versus dose, that's going to have a very helpful impact. Route certainly, matters. We see more inflammation with intravitreal injection than we do with subretinal injection. I estimate it's about a log unit in terms of your dose. So that doesn't mean that subretinal is impervious to it, but just that you can get away with a little bit more vectors subretinally. Suprachoroidal is still, fairly new, so we don't really know what the immune response is as well as we do with intravitreal and subretinal. So immunosuppression prior to treatment is really important. And I think, part of the problem is a lot of times companies will give too little steroids, or they'll taper them too quickly. And what we've really learned is that the inflammation really occurs sort of between 4 and 8 weeks after treatment. And this often corresponds to when the steroids are being stopped. So, one thing that we've tried to do is actually slow the taper of the steroids to make sure that you have coverage during that critical time. We may need to start considering using steroid-sparing agents such as methotrexate or Cellcept. And, this is hard because a lot of ophthalmologists are not comfortable giving these drugs. There's been some data to suggest that rapamycin might actually modulate the immunogenicity of AAV, and I think that's really interesting. And then finally, there are ways maybe that we can modify the vectors so they evade the immune responses. Okay, I'm going to kind of finish up with one last disease, which, I'm very excited about this program. So CEP290 is a ciliary protein that's important for photoreceptor function. When you have mutations in CEP290, you get a very severe form of Leber congenital amaurosis. So many of these patients, when we first see them at 2-3 years old, they have light perception vision. That's it. What's really fascinating, though, is when you look at the retina of these patients, look at the structure of the retina, you can see that there's actually preserved cells in the macula. And we think these are probably cones. So they're present, but they're not working at all. And this is really critical because it means that maybe if we can fix the genetic problem, we could actually restore function. So CEP290 is too big to fit into AAV for a typical gene augmentation. therapy. So the approach has been to do gene editing, and this was Edit 101 was developed by Editas, and essentially this packages a gene editing platform into an AAV5 vector, which is delivered subretinally. And the way this works is that most patients with CEP290 have a common mutation. And it's a mutation that occurs between exon 26 and 27 of the gene and affects splicing. Essentially, it results in a truncated form of the protein that is degraded, so you do not get a fully functional protein. So Edit 101 codes for 2 guide RNAs and Cas9 protein. These then pair up and are basically targeted to flank this intronic mutation and cut it out. And essentially this will give you full-length transcription. So we treated the first patient in Oregon back in 2020. This was very exciting. This is now, we've gone through, treated 14 patients at ascending doses along with our collaborators at Harvard and Penn. And because these patients have really low vision, you have to use different ways of testing. So in particular, most of them can't read an eye chart. So we have to use something called the BRVT, which is a low vision chart that uses gratings to measure their visual acuity. We use something called full field stimulus threshold, which is essentially the patient looks into a big dome, you just flash a light, they click a button if they see it. So it's a measure of global retinal sensitivity. And we also use a maze at different light levels where the patients have to navigate this. So we had a natural history study that defined the parameters that we needed based on the variability, how much change we needed to see, and we went and treated patients. And so here are the results. And so if we look at visual acuity, 4 out of 14 patients actually had an improvement in visual acuity. And this is logMAR visual acuity. So even though there were only 4 patients that responded, you could see some of these patients actually had really incredible improvement. So a logMAR of 1.0 is almost 10 lines on the eye chart. 5 out of 14 had an improvement in FST. 4 out of 14 had an improvement in the mobility. And if we looked at patient-reported outcomes and just asking the patients, how has your life changed? Can you do different tasks? 8 out of 14 actually reported an improvement. If we kind of take a stricter criteria and say how many patients had improvement in 2 or more of these, we actually see that 5 out of 14 did. And even though this is not a 100% responder rate for a phase 1/2 trial in such a severe disease, that's actually pretty amazing. This is just an example of one of our patients. So looking at kind of best corrected visual acuity, you can see they had almost a 0.7 logMAR gain in vision, and this was sustained over time. They also had an improvement in their FST as well as their navigation challenge. And this patient described to me things that were really interesting. So she said that when she used to look out on her porch, it just looked like a single piece of wood. And after the treatment in the treated eye, she could see the individual slats of wood. The other thing was, just at work, she was able to navigate better. And, she said if she dropped something on the ground before, she could never find it. Now she actually had sort of the ability to find something. And so, it really is kind of convincing. This is that one patient showing the mobility. So originally, when you do the mobility test at a very bright level, she failed to complete it. But after the treatment, she was actually able to complete the course at a much dimmer light, once again showing an improvement in sensitivity. So, we're very excited about this. I think it shows that, this was the first gene editing trial in the eye, and it suggests that gene editing can work. And I think that just opens up a whole new frontier for treating inherited retinal degenerations. So in conclusion, in just a short time, we think back to 2008 when the RP65 gene therapy program really started to now, things have just really blossomed. And it's a very exciting time to be in retinal degenerations. It's a very exciting time for patients. And I think we're going to see more and more treatments. Thank you very much.
Thank you, Mark, so much. This was wonderful. And that hope at the end, what you showed, is something we have to accelerate. I have many questions, and there are at least 12 questions from the room, so you're going to be occupied for quick answers. Intravitreal injection and subretinal injection. You remember there was a lot of hype about intravitreal injection is going to be the way to deliver it. Can you comment on that?
Yeah. So the big desire for intravitreal injection really relates to practicality. You can do an intravitreal injection in the clinic. It's very easy. Any retina specialist can do an intravitreal injection. Subretinal surgery is a much bigger deal. You have to take the patient to the OR. Most retinal surgeons are comfortable doing subretinal injections, but it's definitely more work, and you really have to follow the patient post-surgically. So I think from a scalability point of view, it'd be desirable to be able to use intravitreal injections. I think the reality is there have been several problems. One is inflammation, and at least with some of the current vectors, there's sort of this dose response issue where to get to the dose that you're seeing an effect, you're starting to get a lot of inflammation, and that's somewhat limiting. The other issue is penetration. So in a mouse, you do an intravitreal injection, the internal limiting membrane is really thin, and I think the vector goes right through. In patients, there's a much thicker ILM. You can have epiretinal membranes, especially in a disease state. So it's not clear that the vectors really make it to the photoreceptors. And people have tried to make evolutionarily derived capsids. That has improved the delivery. I just don't know if we're there yet. So, I would hope that we can make better intravitreal injections, because ultimately, if we're going to treat thousands of patients, that is probably what we need to do. But I think the technology needs to improve.
Caroline.
Hi, Dr. Pennesi. Thank you so much for your talk. As a physician scientist and a clinical trial specialist, how do you envision that the next generation of genome editing, like prime editing, these different pegRNA molecules will be translated into clinic, especially for neighboring mutations into the same gene?
Yeah. So, once again, I kind of hinted at scalability, right? So we have a big scalability problem. It's amazing that we can treat these patients with RP65, and it can be life-changing for them. But RP65 is maybe 1 in 500,000 patients. As we get to more common diseases, it's going to be very expensive. These treatments are very expensive, and just the logistics of treating that many patients is going to be difficult. So, we need to find better ways to do it. The other, issue is that for gene replacement works great if you can fit, if the protein or the gene is small enough to fit into AAV, but that's not the case. Some of the most common genes are too big. So USH2A, for example, is one of the most common genes for retinitis pigmentosa. It's too big. And it would be great, right, to do, something like base editing, but you have hundreds of mutations. So to design a vector for every mutation is problematic. So that's where I think, the promise of something like prime editing really is attractive, because you could treat, multiple mutations. There's another really interesting technology coming along as well, which is RNA transplicing, whereby you can essentially swap out the 5' half or the 3' half of the mRNA. And that might be another way of, treating multiple mutations without having to design multiple vectors. So there are a lot of cool technologies coming.
Thank you for your talk. So in these trials, there are many constraints, starting from the fact that it's you have these rare diseases, right? So from the point of view of a clinical trial, is there any value in trying actually to allocate resources in chasing patients from a very diverse ethnic background.
Mhm. Well, I think that, certainly when you have rare diseases, recruitment is a challenge. And this is something where I work with companies a lot. And, the company will say, we want a patient with a RP65 mutation who has a visual acuity between 2200 and 2800, has preservation on the OCT, can do a mobility test. And by the time you start taking all these Venn diagrams, you're like, yeah, we have one patient. And so, the recruitment is very hard. So you have to be very careful, what you're doing with rare diseases. I think when you get to more common diseases, it's important to have that kind of ethnic diversity. I think, early on with these rare diseases, we just need to prove that it works so that we can get to an approval. Because if we have approval, then we can go and treat a variety of patients.
This is how FDA thinks.
So for rare diseases, yes.
All right, let's go to Zoom. We have a question from Mike. Mike, are you there?
Apologies. I wasn't sure if you were referring to me. I guess my question is there a need or an argument for concomitant therapy along with gene therapies?
Yeah, absolutely. So, I've always been of the belief that there's not going to be a single silver bullet, for these therapies. You look at other diseases such as cancer, you use every tool in your armamentarium to battle that. And, I don't think gene therapy is going to be the end all be all. And, even right now, you look at the RP65 patients where we've delivered the blood, there has been improvement, but the peripheral retina where the therapy was not delivered continues to degenerate. So, I really think at the end of the day, we're going to see kind of combination therapies. You're probably going to put the patient on some kind of antioxidant or protection. You might give them gene therapy. You might even have cell therapy for delivering, different neurotrophic factors. So I certainly think that will happen. Of course, early on from a regulatory, point of view, it's hard to do multiple therapies. You sort of have to do one at a time, but I don't think that's how we will be treating patients in 15 or 20 years.
Hey, Mark, beautiful and very exciting work. And thank you for this overview of the state of gene therapy. Again, it's very informative. I have two questions. First, following up on what Chris asked about intravitreal versus subretinal. What are your thoughts on the potential of maybe topical or systemic delivery for gene therapy?
Well, certainly if we could do that, it would be fantastic. With, the cornea, they've been able to do topical gene therapy. I think that it's still hard to get penetration back into the retina. With, systemic, you face the issue of the blood-retinal barrier. Now, in retinitis pigmentosa, there is breakdown of the blood-retinal barrier, so it might be possible to deliver. I guess, my question would be that, once again, from an inflammatory, point of view, like how much do you have to give systemically to get it into the retina? It seems like, when they talk about systemic gene therapy, they talk about 10 × 10⁻¹1 per kilogram. And, so I just wonder, is that really the way to go? You're going to have to put a lot of vector in to get to the eye, and that probably isn't going to be favorable.
And my second question is, again, I would love to hear your thoughts about gene-independent therapy for complex diseases such as retinitis pigmentosa.
Yeah, certainly I think we need to develop gene-agnostic therapies. Once again, if we think about, we have 300 different genes. Many of these genes are rare. They're never going to be commercially viable. Maybe the top 15 genes we can develop specific therapies for. But we do need ways of having gene independence. And so we're actually, there has been a trial started for delivering rod-derived cone viability factor as a gene agnostic treatment to prevent cone degeneration. And so that's kind of one of the first ones. There are sort of other ones that are coming down the pipeline as well.
Great, thank you.
Thank you for that really interesting talk. So I was kind of curious for some of the larger genes, like the USH2A, I think, gene. Has anyone looked into like exon skipping therapeutics and whether those would be kind of a suitable alternative?
Yeah.
I was
so-
kind of thinking in like
Yep,
the case of
yeah.
Duchenne muscular dystrophy or something like that.
Absolutely. So yeah, the answer is yes. And so for USH2A, there is a common mutation that occurs in exon 13. And it turns out you don't really need exon 13 for the function. So there's already been a trial using antisense oligonucleotides to elicit exon 13 skipping. That trial showed some promising data. The company ran into some financial issues. It's now been reconstituted and actually it's going to start up again, which is exciting. Similarly, people are looking at using gene editing approaches to just chop out exon 13. So yeah, definitely.
Thank you.
Thank you so much for your fascinating talk. So I have a question regarding the X-linked retinoschisis patient. So I realized that these patients present a very broad spectrum of phenotypes, including aneurysm, telangiectasia, even exudate. So I'm wondering how you choose the injection site and the number of blebs with the subretinal approach. And another question is, have you observed any relationship between the genotype and the benefit of gene therapy in these patients?
Yeah. So for X-linked retinoschisis, at least for this initial phase 1/2 trial, the inclusion criteria are pretty specific. So you can't have any peripheral schisis. You can only have essentially central schisis. And so when determining where to make the retinotomy, they're trying to go outside of where the area of schisis is. And one of the really, innovative technologies that we have is intraoperative OCT. So the surgeon can actually do an OCT at the time of the injection and find an area of retina where there's not schisis and deliver it there, and then try to limit the bleb such that you're not going into that area of schisis. So that was question one. What was the second one?
So is there any relationship between the genotype and the benefit of the gene therapy?
Yeah, I think it's too early to say. With RP65, it's a rare disease, and most of those patients are loss of function. There are hypomorphic. There are some mutations that are hypomorphic, and I will just say from my own personal experience of treating these patients, the patients who are hypomorphs don't seem to have as much of a benefit. But I think maybe that's because it's kind of a ceiling effect already. You have some patients who have no functional RP65, and when you put the gene back in, it's like turning the light on. I mean, they have a huge 4 log-fold change in their sensitivity. With some of these hypomorphic mutations, they might only be down a log unit or two, and so I think the difference may not be as much.
A great talk. I have a question regarding the inflammation. So, is there any clue on what is causing more inflammation, DNA from AAV or the capsid? And second question, are there any ideas of humanizing the capsid, attaching some engineering the capsid so it has some human proteins, but it's not being triggering the inflammation so much, but still enabling the virus to get to the cell?
Yeah, I mean, I think that you're probably getting outside of my expertise domain. There have been studies, and some of these have been published where they've taken empty capsid versus full capsid versus partially full capsid, and you do get sort of different inflammatory profiles, and certainly the CPG content in the DNA can influence it, but one of the difficulties is every company has different manufacturing processes and different process residuals. And so it's kind of hard to do a head-to-head comparison. There are some people that are looking at this, and certainly there are ways of engineering the capsid, to put kind of like an anti-TLR9, motif in, but I don't know about like humanization.
We have a question from Zoom. Steve?
Mark, Steve Fleisler in Buffalo, terrific talk and very illuminating. So my question was about why nanoparticles have not really advanced from the proof of concept in animals model studies. They have advantages of lack of immune response and no limitation of cargo size.
Well, I would say wait until next year, because there's a whole lot going on in the LNP space right now. I think part of the difficulty, some of it might have just been you needed time for the technology to catch up to the idea. But I've seen a lot of data now showing the ability to take LNPs and target photoreceptors with subretinal delivery. There's sort of difficulties in that you can design an LNP to use sort of like an evolutionary approach, right? And there are a lot of ways you can modify the lipids, many different options, and people have used sort of a barcoding, approach, sort of the same way you can evolve an AAV to find LNPs that will target photoreceptors. Part of the problem is that you find that one that works, and say you edit GFP or something. When you go then to put your other machinery in, it changes the shape and properties of the LNP, and so then you might lose your specificity, to getting to the photoreceptor. But I actually, think, like I said, I've seen a lot of encouraging data in the last year, so I think we're going to see that happening very soon.
Hello, great talk. Great talk. I was wondering about this question, I guess, is a follow-up to Dr. Fleisler's question on a lot of what you presented is AAV mode of delivery, and gene augmentation approach. But you also showed that there's other ways of gene editing and gene silencing, and I was wondering how close some of those other approaches are along, and other delivery modes are along.
Yeah, I mean, there is so much in the pipeline, and literally about once a week, I get a call from a company that has new technology that they're working on. So we're getting ready to start a clinical trial for a dual vector approach that's using, NTNs to essentially splice together, larger genes. I sort of alluded to, there's another company working on RNA transsplicing. So there are just many, many things coming. So it's exciting. It's really exciting.
Hi, Dr. Pennesi. It's a very nice talk. I enjoyed it. So I'm a pharmacist, and sorry, this is a non-scientific question, but I think about the logistics of getting drugs, and I wondered how you deal with the risk of receiving, a $500,000 drug, and how you insure against that, and how it all works.
So we're very, being at a university, we're very fortunate to have a robust research pharmacy, and with a lot of infrastructure. But when we were, first starting to do this, we did a lot of dry runs. And for example, I don't know if you know the geography of OHSU, but the hospital's up on a hill, and the research pharmacy is at the bottom of the hill, and they're connected by this tram. And so, when you get the product, it gets prepared at the research pharmacy, and they got to put it into a cooler, and then you have to go up the tram, to take it to the OR. And so, we had to do all these kind of practices to say okay, how much time is it going to take to, get up and, well, what happens if the wind is 30 miles an hour and they close the tram and now we've got to drive it up, so there is a lot of planning that goes into that. Similarly, do you have a backup -80 freezer, because you don't want to lose millions of dollars of product. And so being at there are definitely advantages of being at a big, university that has that infrastructure. The other thing was when Luxturna, came along, the university formed kind of a high value drug, committee, because we sort of knew that gene therapies were going to be coming, and they thought a lot about, how are we going to deal with these things, and make sure that they're protected.
Hello, thank you for nice time. As a vitreoretinal surgeon, I have a question. As you know, retina is has ample surface, and each vector, I think, it can infect one cell. Is it right or not? And how can you be sure that you can deliver the vector to all diseased cells?
Yeah. Well, so first off, in full disclosure, I'm not a vitreoretinal surgeon. That credit goes to Andy Lauer, who's done most of our surgeries. But the reality is, at least with the current surgical techniques, we can't treat the whole retina. In fact, we have had some experience trying to treat peripheral retina, and that's not a good idea. You can actually get a lot of issues in terms of the vitreoretinal interface and contractions. So primarily we've targeted macula or sort of around the macula. You can make multiple blebs. So some of it depends on just how much product you have. Typically you're getting 200 to 300 microliters, and so people are starting to try different approaches of do we just treat the macula? In some diseases, maybe you don't want to treat the fovea. Can you kind of make a ring of blebs? But I think, very much, we're still learning. Similarly, there can be issues when patients, many of these diseases, the patients have high myopia and they can have staphylomas, and that can create issues when you're making the subretinal bleb.
I want to know, each injection can affect how much surface of retina?
Mm-hmm. It's hard to know because you don't really have great control of the formation of the bleb. And, some blebs form and they're kind of like these very high peaked blebs, with a small area. And other times you get these very, broad, shallow blebs, but you don't really have control over that. Really all you can do is, go or not go. And, so a lot of times, they've sort of switched over from doing injection to using the foot pebble for, an infusion. And that lets you go very slow. And in fact, there is a relationship to how fast you infuse and maybe damage to the RPE. So there's sort of a threshold. So a lot of times, the surgeon may, put in some and kind of watch it and then, go a little more. And then if you say, I don't like the way that bleb's going, you stop and maybe make another, a second bleb.
Yes. Hello. Thank you for the talk. You mentioned that rapamycin might modulate the immunogenicity of AAVs. Is this efficacy only applicable within a certain time frame? And why or why not might that be?
Yeah, I'm going to say that I'm not an expert on that, so I don't know the answer to that.
Hi, Dr. Pennesi. Thank you so much for this talk. When thinking about responders and non-responders in a clinical trial, can you comment on maybe, I don't know if I'm using this term correctly in the space of clinical trials, but kind of the positive predictive or prognostic factors that you can look at to kind of parse out what makes somebody a responder versus a non-responder?
That is a great question. And it's probably about an hour answer. So
go ahead.
Yeah. Well, I think you have to work a little backwards here, right? And really say, well, what defines a responder? I think there is what the patient would consider important to them and what they would define, as being success. There's what the regulators would define as being success, and then there's what the investigators might, define as being success. And those can all be different answers. So, the difficulty is that we need to get these therapies approved so that they can be delivered to a larger number of patients. That often means that in a trial, you're picking sort of the Goldilocks patients. You're looking for kind of the perfect patient because you need to show an effect. Because if you don't, the program's going to end. And there are many promising programs that have never made it to approval, for a variety of reasons. So working with the FDA is challenging because they don't really always tell you the answer as to what they want. They often say, come to us with your data and then we'll let you know. We have some ideas of what kind of things that we need to show to the FDA. So, they really want to know what is clinically meaningful. And so there's certain things like, for example, a gain of 15 letters on the eye chart will not be contested. If you can show that you improve visual acuity by 15 letters, you definitely will get approval. It gets more difficult when you're dealing with patients with retinitis pigmentosa because many of those patients may have good vision, right? They may have 20/20 vision, so they're not going to be able to improve, 15 letters. So then you start getting into these visual field measurements and what is, clinically meaningful, visual field improvement. And some of the data comes from glaucoma and, there's sort of these very specific criteria about, well, you need to get a 7 decibel improvement at 5 points. So we kind of try to work backwards from what we know the regulators want and try to pick patients where we think that their vision is not so bad that, they won't improve and not so good that they're going to, have a ceiling effect and find patients that are sort of in the right, range. So that's one hurdle. But then, even after FDA approval, then you have the whole insurance payer world, which is you can get a therapy approved by the FDA, but that doesn't mean the insurance is going to pay for it. And there you need to really show more, a benefit to the patient in terms of their quality of life. And so we have to look at things like patient-reported outcomes, which are sort of these long questionnaires about, how hard is it for you to, find something in the dark or, navigate in a movie theater. And we have to kind of make measurements there to show that you're actually impacting the quality of life. So, there are, several different hurdles that you get through. And so you can kind of back extrapolate in a trial to figure out, okay, who do we think the best patient is? But it's not easy.
All right, we had a great conversation. Last quick 3 questions, and we'll let you be.
Hi, Dr. Pennesi. Really enjoyed your talk. In terms of measuring clinical outcomes, you had shown the ellipsoid zone analysis from OCT imaging. Could you comment on how that compares to some of the other standard measures, microperimetry, for instance, in terms of sensitivity, throughput?
Yeah. So we know that psychophysical measurements are generally more sensitive than structural measurements, at least right now. That may change in the future. But so to have a defect on the ellipsoid zone, you usually need to have about an 8 decibel drop in sensitivity on microperimetry. Once that happens, you often have this transition zone. So you'll have intact ellipsoid zone, and then outside you'll have where the ellipsoid zone is disrupted. If you map the microperimetry on top of that, there is a pretty good correlation. But you lose some information, I think, with ellipsoid zone because it's sort of a binary measurement. It's there or it's not there. Whereas with, sensitive microperimetry, you're getting a little more of a gradated, answer. So you can have areas where the EZ is still intact, but the microperimetry sensitivity might be down. Now, there probably is a lot more information in the structural data. So, you can look at, there's actually information encoded in the brightness of the ellipsoid zone, and people are starting to look at that. It's a little bit tricky to measure that because you need to normalize it. But also there are other really interesting optical ways of looking at the photoreceptors. So you can measure the outer segment length. There's something called optoretinogram, where you can look at the motion of the outer segments in response to light. So it's almost like an optical form of an ERG. So there are a lot of kind of newer technologies coming along where I think, we might be able to improve our ability to assess function.
Hi, Mark. That was really optimistic and empowering talk. I have a question regarding the inflammation again, and actually use of steroids. As you mentioned, this is not favorable. And I still wonder why there is no use of direct inhibitors of, the response to viruses, so innate immunity, instead of GR. I mean, instead of steroids that inhibit only part of the response.
Like when you say antiviral?
No, more like, because this is an FKB signaling pathway usually, so anything that will affect the, interleukin-1 receptor or other pathways that feed into. And GR is only on the side and it's delayed.
Yeah, so I mean, this is something we talk about a lot. My colleague at Oregon, Paul Yang, is uveitis trained. And, so whenever we have inflammation, I call him in, and, he often does end up putting patients on different, steroid sparing agents, because the problem is, well, I would say steroids work really well because they just sort of suppress everything. But the problem are the side effects that you get. So after about 2 or 3 months on steroids, the patients start gaining weight. They have a lot of other, they can have systemic, they get cataracts. So we have used some, immunosparing agents in certain situations. I will tell you that many in a kind of a sponsored driven trials, the sponsors are very reticent to pull out those kind of big guns, partly because, as I mentioned before, a lot of people are not comfortable prescribing those agents. But I do think that we need to consider that in the future. It'd be nice to not have to use them. I'd rather all the smart PhD scientists here figure out ways to make these vectors less inflammatory, but we may need to use steroid sparing agents.
You started with some, and we will finish with
Okay.
Thanks, Dr. Pennesi. So I wanted to ask about the pool of patients available for these therapies. Is there any concern that having so many clinical trials will kind of unnecessarily split up the patient pool? And B, if these trials fail, can these patients ever receive another therapy, or are they just kind of out of luck?
Yeah, that's a great question too. I remember really thinking about this the first time we had 2 trials for a given gene, and I kind of went into the literature to do some research as to, well, what do they do in other fields like cancer or other rare diseases, and there's actually a really interesting set of literature, kind of ethical debates of how do you pick, let's say you have a very small population of patients and more than one treatment, should the doctor just decide which patient, or which treatment the patient gets? Should the patient decide? Should you randomize it? And, there's sort of arguments both ways. Like one argument would be that if you kind of split up the population, both trials may fail because they may fail to recruit the necessary, number of patients, and that maybe a paternalistic model is actually better to say, this patient's going to go to this, trial. And then, there are counter arguments, that say, well, patient autonomy is important, and, people need to be able to decide, which trial they go into. So I don't know if there's kind of a resolution. I think for us, fortunately, sometimes it works itself out. A lot of times, because each trial has its own inclusion/exclusion criteria, patients, might only end up being eligible for one trial. And, so that was like with X-linked RP, at one point we had 3 different trials for X-linked RP. But one of them, said, well, we only want patients with rod cone dystrophy. We're going to exclude any patients who have cone rod dystrophies. Another one said, we only want mutations in, OR15. We don't want mutations in exons, 1 through 14. So it hasn't, on a practical level, I don't think it's occurred yet, but, we try to be very open with the patients to say, here are the different, treatments. We don't know, if one is better than the other, here's what we know. And some patients are highly educated and they actually go and pull the papers and say, I want to be in this, trial. Other patients are like, just decide for me, I trust you. So, and that's kind of part of being a doctor. I mean, that's a little bit of part of the art of medicine is, kind of figuring out how to deal with these situations. So it's a really great question.
Mark, thank you so much. You saw how many questions? We had twice as many.
Yeah, it's great.
But we will torture you during your day visit.
Sure.